Hello students, so welcome back to the channel. In this video we are going to study about the internal structure of root, both dicot and monocot root. So first let me explain you about dicot root. See, in the dicot root, outermost layer, which is known as epiblemma or piliferous layer, it is not covered by cuticle and it does not have even stomata also. Okay, so this is the outermost layer which is known as epiblemma or piliferous layer. And it is also known as rhizodermis. So these are the different names of the outermost layer. It will not have cuticle will be absent and stomatas will be absent. So I'm just writing it here, try to understand. Cuticle is absent and even stomatas are absent. But they are having the presence of root hair. Root hairs will be present in them. So this structure will be present in the root anatomy. Now hypodermis is absent in the root and cortex is entirely parenchymatous. Is it clear? Inside this, after this you are finding the next layer is your out inside to the cortex. Okay, but inside to the cortex you are finding this is your endodermis. So this layer is known as cortex, which is made up of parenchymatous. This is cortex. And then next layer, this is your endodermis. Inside to the endodermis will be pericycle. This is pericycle present inside the endodermis. And inside the pericycle, vascular bundles will be present in the radial arrangement. Because we have already studied that roots are having radial arrangement of vascular bundle. Radial arrangement means one radius xylem, one radius phloem. Is it clear? So this is xylem present. This is also xylem. I'm drawing for xylem. And xylem is with the exarch condition. Exarch condition means protoxylem is facing outside. And this is phloem. Okay, this is phloem. This patch which I'm drawing, this is phloem. So radial arrangement of vascular bundle is present. Now in detail, we'll be doing one thing. We'll be studying this part and then we will study it in detail. Check my diagram now this side. See, this is your epiblemma or piliferous layer. Okay, which is only one layered. And the cell which will give rise to root hair. The cell which gives rise to root hair, that cell is known as, just 1 minute, beta. This is the cell which is going to give rise to root hair. So this cell is known as trichoblast. Okay, this cell is known as trichoblast. The cell which give rise to root hair, this is known as trichoblast. And root hair are unicellular and they are epidermal outgrowth. So this is your piliferous layer. I hope this much labeling is clear to you. Is it clear? Next, I am drawing the labeling of the endodermis in circle. This is endodermis. We have to draw in the circle because we are studying complete. This is the cells which I am drawing it in this endodermal cells. And between the epidermis and endodermis, this is your cortex with loose parenchymatous tissue. Okay, this is the cortex which is present, which is loose parenchymatous tissue. And their loose reason is they have to main work of the root is to absorb water and mineral. So water and mineral has to reach to the xylem through intercellular spaces also they can reach, and from cell to cell connection through plasmodesmata also they can reach. Is it clear? That is why they are having loose intercellular tissues, and they are parenchymatous only, so that apoplast or symplast movement of water can be achieved. And this topic we'll be studying in detail in transport chapter. This much is clear, beta. Now see, let me draw the endodermal layer. So now I'm drawing the endodermal layer. Just a sec, color changing is taking time. This is endodermal layer now. Sorry, pericycle layer. Endodermal already we have drawn with the blue color. This is now pericycle layer. This is pericycle, green color which I have used for pericycle. And now see, this is your xylem. This is xylem with exarch condition. Okay. Radial arrangement we have to remember, radial arrangement with exarch condition, and it is showing centripetal growth. Centripetal growth you know very well. Metaxylem is moving towards the center. Clear beta? Metaxylem is moving towards the center. So this is the way how the xylem is growing. So centripetal growth is there. Now you have to focus carefully. This is your phloem. I'm using this color for the phloem. This color I'm using for the phloem. This is the phloem patch. So one radius xylem, one radius phloem is present. Now check the board carefully. The cell of endodermis, just opposite to the protoxylem, this cell, okay? This cell and this cell and this one. Total 4 cells. Are you understanding? Total 4 cells will not have deposition of Casparian strips, means suberin, but remaining cell will have the deposition of the suberin in the form of a Casparian strips. Please, it is very important, try to focus it. Ki remaining cells will have the deposition of suberin in the form of Casparian strips. Only the 4 cells which are opposite to the protoxylem will not have Casparian strips. And these 4 cells, let me highlight with the white color so that it becomes easy for you to understand. These 4 cells, okay, but these 4 cells, okay, they are known as passage cell. They are known as passage cells because the water can enter through this passage cell only into the xylem. Is it clear? So these are your passage cell. Please highlight it carefully, understand carefully. Passage cells are also known as transfusion cells. So these are your 4 passage cells, and they will be always present just opposite to the protoxylem. Is it clear? Now, see carefully the tissue which is present between the xylem and phloem. This is known as conjunctive tissue. I am doing the filling with the white color. The tissue which is present between the xylem and phloem is known as conjunctive tissue. Please understand the concept carefully. We will not say pith or medullary rays. No, they are conjunctive tissue and they are very small parenchymatous cell. They are very small parenchymatous cells. So parenchymatous cells which is present between the xylem and phloem is known as conjunctive tissue, and please remember it is not known as pith or medulla. Clear? Just give me 1 minute. Let me cover this area. And now see this part, center path, this one. This is known as pith, which I've highlighted with blue color. If you see the diagram, pith is highly reduced or it can slowly become absent also. Understood the anatomy diagram? So let me do the labeling. This is your cortex layer, which I have drawn it. This is your cortex layer. This blue color is your endodermis. Okay. This blue color is your endodermis. And then green color, this is your pericycle. This is green color is your pericycle. And then the white one, which is present between the xylem and phloem. This white one, I'm writing it, labeling here. This is known as conjunctive tissue. And this blue color in the center, this I'm just doing the labeling here. Please try to understand. This is known as pith or medulla. Now I want to explain you here. Please try to understand one concept. Why they are having this arrangement of the loose parenchymatous tissue. I'm using one color. Please try to understand that thing. Okay, just give me 1 minute. See, the water is absorbed by the root here. Focus the color. It can come through intracellular spaces. Are you able to see? It is coming through intracellular spaces and finally it is going to the passage cells and then water is coming to the xylem. Understood the diagram? Or see carefully what I will do. I'll use another color. Water can come, check the color please. Water will come from cell to cell. See, water is coming from cell to cell and finally again to the passage cell, pericycle, and is given to the xylem. So if suppose water is coming from cell to cell, then this is known as symplast pathway. I'm writing it here. This is known as symplast pathway. But if suppose water is coming from intracellular spaces, this one red color which was coming from intracellular spaces, this is known as apoplast pathway. Apoplast pathway. So I hope this diagram is clear to you. Now this is a dicot root and same diagram will be of monocot root. Then what is a basic difference? Let me highlight it. Class, the basic difference between the dicot and monocot root is, if you see, they are having both endodermis layer, which is having the Casparian strips in the form of suberin. Okay. So they are having highly suberized Casparian strips. Then they are having pericycle. But if you check the difference, you will find the differences with respect to the number of the vascular bundles. Is it clear? So here we will say vascular bundles are 2 to 6. And here we will say vascular bundles are more than 6. Because 2 to 6, so we will say they are diarch to hexarch. Diarch to hexarch. And here we will say they are polyarch because they are more than 6. And as a result, if you see in the diagram, clearly you will find here pith is well developed. Pith is well developed. But in case of the dicot root, we will say pith is highly reduced or absent. It can be absent also, okay? It is highly reduced or absent. So this is your xylem, okay? All this which I have used, this color, this is for xylem. So if you see here, the xylems are more in the monocot root, but in case of the dicot root, their vascular bundle I have drawn only 4, but it can go maximum only up to 6. Okay, and this pink color is for phloem. This is phloem which I have drawn it. So same number of xylem and same number of phloem should be there. And this white chalk, you know very well, this white chalk is the conjunctive tissue. So conjunctive tissue is present between the vascular bundle in the root anatomy. So I hope this much topic is clear to you. Difference between the dicot and monocot root with respect to the internal structure. This topic is clear. Thanks for watching.